Genetics is the branch of biology that covers the study of heredity and variation. Trait. A trait is any heritable feature of an organism. Traits include physical features such as hair color, biochemical features such as blood group, behavioral features such as anger. Traits are controlled by genes. Genes. Genes are the units of heredity. Chemically, they are portions of DNA on the chromosomes. Mendel's factors are equivalent to genes. Mendel represented gene by a letter. For example, the gene for plant height in the pea plant is represented by the letter T. Alleles. Alleles or allelomorphs are alternative forms of a gene located at a specific position on a homologous pair of chromosomes. For example, the gene for plant height in pea plant has two alleles. The allele for tallness, represented by capital T, and the allele for dwarfness, represented by a small t. The paired factors described by Mendel are equivalent to alleles of a gene. Homozygous condition. If the alleles of a gene present on homologous chromosomes are identical, the condition is said to be homozygous. For example, a pea plant with both homologues carrying an allele for tallness will be a homozygous tall plant. Similarly, a plant with both homologues carrying the allele for dwarfness will be a homozygous dwarf plant. Heterozygous condition. Heterozygous individuals have different alleles of the gene located on their homologous chromosomes. A pea plant with two contrasting alleles for plant size, one for tallness and the other for dwarfness, is an example of a heterozygous individual. Dominant allele. Of the pair of alleles, the one that expresses itself whether present in a homozygous or heterozygous condition is the dominant allele. For example, pea plants are tall in homozygous and heterozygous conditions. This shows that the allele for tallness is dominant over that for dwarfness. Dominant alleles are generally represented by a capital letter. Recessive allele. Of the pair of alleles, the one that is not able to express itself in the heterozygous condition is the recessive allele. For example, the allele for dwarfness is recessive. It is not able to express itself in heterozygous pea plants, but can express itself only in the homozygous condition. Recessive alleles are generally represented by a small phenotype. Observable or measurable characteristics constitute the phenotype of an individual. For example, the phenotype of both these two pea plants is tall, whereas this pea plant is dwarf. Genotype. The genotype is the genetic makeup of an individual. For example, these two pea plants are phenotypic tall, but the genotype of first plant is capital T, whereas the genotype of the second plant is capital T, small t. The first plant is homozygous and the second is heterozygous. Emasculation. Emasculation is the process of removing stamens from a flower before dehiscence of anthers to prevent self-fertilization. It is done in a flower that is considered as a female parent. Artificial hybridization. Artificial hybridization is the process of artificially crossing two genetically dissimilar parents to produce hybrids. For example, a cross between tall and dwarf plants. Hybrid. A hybrid is the organism produced by crossing two genetically different parents. For example, in a cross between tall and dwarf pea plants, the progeny produced are hybrids. P generation. In a hybridization experiment, the P generation is the parental generation. For example, in this cross, tall and dwarf pea plants are the parents, or the P generation. F1 generation. The first filial or F1 generation is the first generation of offspring produced from a cross between genetically dissimilar parents. For example, in a cross between a tall and a dwarf pea plant, the heterozygous progeny belong to the F1 generation. Selfing. Selfing or self-fertilization is the process in which plants are allowed to self-pollinate. This is also called inbreeding. F2 generation. The second filial or F2 generation is the second generation of offspring produced from a cross between the genetically dissimilar parents. For example, in this cross, the progeny produced by the selfing of the F1 generation belong to the F2 generation. Punnett square. A Punnett square is the grid devised by R.C. Punnett, used to view the gametes of each parent and the possible combinations of offspring in a cross. For example, in this cross, both heterozygous tall parent plants will produce two types of gametes, one with the dominant allele, capital T, and the other with the recessive allele, small t. As you can observe, with a Punnett square, it is easy to work out details of the possible progeny. Monohybrid cross. A cross between two individuals which is performed to study the patterns of inheritance of a single pair of alleles for one characteristic. For example, a cross between purple-flowered and white-flowered pea plants to study the patterns of inheritance of alleles for flower color is a monohybrid cross. Mendelian monohybrid phenotypic ratio. The 3:1 ratio of phenotypes of F2 progenies obtained in a monohybrid cross is referred as the Mendelian monohybrid phenotypic ratio. For example, if the purple-flowered plants of F1 are self-fertilized, the F2 generation will have purple and white-flowered plants in a ratio of 3:1. Back cross. A cross between a hybrid and one of the parents is called a back cross. For example, a hybrid produced in a cross between a white and a purple-flowered pea plant. A back cross will involve crossing this hybrid with either a white or a purple-flowered parent plant. Test cross. A cross between a hybrid and the homozygous recessive parent is called a test cross. For example, a hybrid produced in a cross between a white and a purple-flowered pea plant. A test cross involves crossing this hybrid with a homozygous recessive white-flowered plant. In a test cross, dominant and recessive progenies are produced in equal proportions. In other words, the ratio of dominant to recessive progenies is 1:1. A test cross is performed to find out whether an individual is homozygous or heterozygous dominant. Reciprocal cross. A reciprocal cross involves performing a cross with a reverse combination of parental sexes for the same characteristic studied. For example, if the first cross involves crossing a male white-flowered pea plant with a female purple-flowered plant, Then the reverse cross will be a cross between female white-flowered plant with a male purple-flowered plant. Dihybrid cross. A cross between two individuals which is carried out to study inheritance patterns of two pairs of alleles controlling two different characteristics is a dihybrid cross. For example, a cross between a round and yellow-seeded pea plant and a plant having wrinkled and green seeds is a dihybrid cross. Mendelian dihybrid phenotypic ratio. The 9:3:3:1 ratio of F2 progenies obtained in a dihybrid cross is referred to as the Mendelian dihybrid phenotypic ratio. For example, this F1 heterozygous yellow and brown seeded pea plant on selfing will produce progeny in a ratio of 9 yellow round, 3 yellow wrinkled, 3 green round, 1 green wrinkled.